For this video today, we'll be looking at linkage by test crossing an F1 generation with the homozygous recessive to see the linkage between genes. So for this example, it's about black bears, where round ears are dominant to pointy ears, large skulls are dominant to small skulls, and black fur is dominant to cinnamon fur. So I took a round-eared, small skull, black fur bear and mated it with a pointy ear, large skull, cinnamon fur bear in order to create this F1 generation. I then test crossed it with the homozygous recessive and observed all of the following progeny. So I first looked and found out which ones would be the NCOs and the DCOs in order to figure out what would be located in the middle for when I created the map. This allowed me to determine which ones would be the SC1s and the SC2s. As you can see here, I've determined that the ear shape would be on the left, skull size in the middle, and coat color on the right. I just want to note that the math for figuring out if this is due to independent assortment or if it's due to linkage would take the whole time of the video to cover, so I'm just going to move forward with the understanding that the chi-square analysis of each phenotype showed that it's not due to independent assortment, but it is due to linkage. So now that I figured out which gene would be in the middle by comparing the NCOs and the DCOs, I had to create the map, which you can see here with the non-recombinants on the top and the bottom. I calculated the distance in centimorgans by figuring out the total amount that crossovers occurred and divided that by the total for each region, giving us the values of 21 centimorgans and 25 centimorgans. So I used this data to calculate the coefficient of coincidence by taking the number of the observed DCOs and dividing it by the expected number of DCOs. I found the expected DCOs by taking the recombination frequency of R1 and R2, multiplying them together, then multiplying that by the total amount of offspring. So after taking the observed number and dividing it by this value, we get a coefficient of coincidence of 0.95. I used this coefficient of coincidence to calculate the probability of the double crossover event occurring, then multiplied that by 0.5 in order to determine the probability of a specific gamete being produced. To calculate the probability of a single crossover occurring, or a specific single crossover occurring, I took the recombination frequency of R1 and subtracted the probability of a double crossover event occurring, which you can see here. And I also multiplied that number by 0.5, since there can be two different single crossover gametes, which gives me a value of 0.08 or 8% for a specific single crossover gamete. And finally here, we can visualize what it would look like on each chromosome for each of the events to occur for every possible genotype.